For our last class discussion in microbiology lab with our differential and selective media, we are going to discuss our three most complex media we use in lab. We will discuss SEM, TSI, and MRVP. Before we can get into these types of media, I need to introduce to you a new media type we have not encountered before. Up until this point in the lab, we have encountered a broth, which is a liquid in a test tube, a plate, which is a petri dish containing a solid media, and a slant, which is a test tube containing a solid media. Today, one of our media is going to be a semi-solid. This is a media that, when applied to a test tube, is too solid to be able to be shaken and move around like a broth, but it is too liquidy to hold that slanted appearance we see with our solid agar placed in a test tube. The way we know it's motility media is we look for the flat top that is not liquid in the test tube. Semi-solid media is used to tell us motility. This media is inoculated with a needle instead of a loop. The bacteria on the end of the needle is dipped straight down the center, not touching the bottom, and then pulled straight back out. The bacteria are applied to the center. If the bacteria are not motile, they will only be able to grow in the center region where they were applied. If the bacteria is motile, you will see a fuzzy outgrowth or cloudiness throughout the media. Our first media to discuss today, SEM, allows us to test for three completely different and unrelated characteristics. The S stands for sulfur reduction, the I stands for indole production, and the M stands for motility. The fact that this media tests for motility tells us that this is going to be a semi-solid media. Let's first start with the sulfur reduction. Sulfur reduction occurs when a bacteria has the ability to reduce or convert sulfur into something called hydrogen sulfide. Hydrogen sulfide then combines with iron present in the media, forming some compound that occurs as a black precipitate. A precipitate is a small solid in the mixture. What this boils down to is if a bacteria is added to SEM and it is capable of reducing sulfur, we will get this characteristic black color shown here in this test tube containing Salmonella typhimurium. The indole production occurs only if a bacteria contains the enzyme tryptophanase. Tryptophan is an amino acid found in many proteins. Some bacteria have the ability to break down this amino acid as a form of nutrition. The bacteria that can break down the tryptophan produce a chemical called indole. Indole itself is a clear liquid. Therefore, after we allow our bacteria to incubate in the SEM media for at least 24 hours, we add a mixture called Kovacs reagent, which will turn the indole a red color if present. Now, putting the two things we've already discussed together, if we look at this test tube here containing Streptococcus pyogenes, it is still just a plain yellow color. Therefore, this bacteria cannot reduce sulfur or produce indole. This test tube here contains E. coli. We see a red layer on the top, no black. So this bacteria does not reduce sulfur, but it does produce indole. This test tube, Salmonella, reduces sulfur, does not produce indole. Proteus vulgaris does both. As far as determining whether our bacteria is motile or not, we simply look for cloudiness in the media. This is not something that we can really do by looking at a picture through the computer. This is the reason that we will inoculate the media in lab so that you can see the cloudiness yourself. Our second media for the day is called TSI. This stands for triple sugar iron. Triple sugar iron contains 3 sugars, lactose, sucrose, and glucose. Ferrous sulfate, which is iron, allows us to check for sulfur reduction, and the pH indicator phenol red. We have used or discussed all of these components, so you should be able to predict some of the results. This is just a complex media. Let's first draw our attention here to the solid red slant containing Micrococcus luteus. It is still red. We've learned previously that if phenol red stays red, that indicates no sugar use. Next to it, with the salmonella tube, we see red and black. This again tells us no sugar use because it stayed red, but we see the black, so that indicates sulfur reduction. Moving here to the S, the Staphylococcus aureus tube, it turned completely yellow. That tells us that the phenol red pH decreased because the bacteria were capable of fermenting or using the sugars. Now moving here to this last tube, Bacillus megaterium, we see a red and yellow mixture. This tells us that the bacteria was only capable of fermenting the glucose, just one of the sugars. Now I draw your attention to this first test tube. This first test tube has a red and yellow media that has been pushed up the test tube. What could possibly be produced in the test tube to cause the agar to rise? The answer is gas. Therefore, this test tube tells us that this bacteria can ferment the glucose only, and while doing so produces gas. Our last media, MRVP, stands for Methyl Red-Voges Proskauer. This is a media that is specifically designed to differentiate between gram-negative intestinal bacteria, or more commonly called coliforms. It is typically only used if you are trying to differentiate between E. coli and Enterobacter, two of the most common gram-negative intestinal bacteria. The way this works, you inoculate 2 MRVP tubes at the same time and allow them to grow for at least 24 hours. 24 hours later, to one of those test tubes, you complete the MR test. This is a test for mixed acid fermentation. If mixed acid fermentation occurs, then you will see a red color form. So here's what the media looks like with nothing happening. Here's what the media looks like with E. coli, Enterobacter, and Serratia after the MR reagents have been added. Notice E. coli is the only one that is MR positive, shows the red color. To the second test tube, we would add the VP reagents. These reagents test for the production of 2,3-butanediol. This is a fancy way of a bacteria to break down glucose. If the bacteria is VP positive, you would see a red color. So here we have uninoculated media, Enterobacter aerogenes, and Escherichia coli. Notice this time the Enterobacter is the one that turned red. It is VP positive. So what eventually you end up looking for is if your MR turns red, you have E. coli. If your VP turns red, you have Enterobacter. If neither turn red, you do not have a gram-negative intestinal bacteria.